Welcome to the Deep Dive, where we cut through the noise of complex topics and distill them into essential knowledge just for you. Today, we're embarking on an incredible journey, a true deep dive into the precise and really rapidly evolving world of genomic medicine and the gene therapy program. This is a field that quite literally promises to rewrite the very code of life to fight disease.
It really does. And you've provided us with a fantastic collection of sources detailing these, well, intricate mechanisms and truly groundbreaking applications of gene therapy. Our mission today is to unpack these complex strategies, reveal some of the surprising facts behind how scientists are literally editing life's fundamental blueprint, and help you truly understand the why and the how of this cutting-edge medical revolution.
Yeah, we'll explore everything from fighting cancer at a genetic level, which is huge, to correcting inherited disorders like Crigler-Najjar syndrome, which we'll definitely dive into later. Get ready for some serious aha moments, because by the end of this deep dive, you'll be remarkably well informed about a medical frontier that is transforming healthcare right now. So let's unpack this by starting with a really formidable adversary, cancer itself. We all know the name, but at its most fundamental level, what exactly is cancer?
Right. At its core, cancer is defined by the uncontrolled, abnormal division of cells. Its growth gone wild, essentially. When these rogue cells accumulate, they form a mass, which we call a tumor. Now, it's really crucial to understand there are two main types of tumors, benign and malignant.
Okay.
Benign tumors are typically localized, they tend to stay put, and generally don't invade neighboring tissues. Malignant tumors, however, are the ones we truly associate with cancer. They don't just show uncontrolled division, but they also possess this dangerous ability to invade surrounding tissues and spread to other parts of the body through a process known as metastasis.
Metastasis, yeah.
Yeah.
That's the really scary part, isn't it? The spreading.
Exactly. And it's not just one disease, as you hinted. Cancer has many, many faces, many origins.
Right? It seems incredibly diverse.
Absolutely. And that incredible diversity stems from its origin point in the body. For instance, carcinomas are cancers that arise from epithelial cells. These are the cells that line surfaces of the body and internal organs. Think of, breast, colon, prostate, or lung cancers. Those are common examples. Then you have sarcomas, which develop from connective tissues, like bone sarcoma or maybe hemangiosarcoma, which affects blood vessel linings.
So vastly different starting points, even at that basic tissue level.
Precisely. And moving on, lymphomas and leukemias are cancers that originate from hematopoietic or blood-forming cells found in the bone marrow and lymph nodes. Germ cell tumors are derived from pluripotent cells. Those are cells with that remarkable ability to develop into many different cell types. These often appear in the testicle, which would be a seminoma, or the ovary, called a dysgerminoma.
And what about those more unusual names like blastoma or adenoma? They sound different.
They do. Blastomas are cancers that come from immature precursor cells or embryonic tissue. A classic example is retinoblastoma, which affects the retina, usually in young children. And finally, we have adenomas, which are benign tumors of glandular origin. If an adenoma becomes malignant, it's then called an adenocarcinoma. So that distinction between benign and malignant is absolutely critical for understanding the disease's progression and, naturally, how we approach treatment.
Okay, that makes sense. It sounds like a cascading series of cellular missteps. Our sources had this really clear schematic, almost like a flow chart of things going wrong. Could you walk us through that journey from a normal cell to a full-blown tumor?
Certainly. It really illustrates the multiple failure points, the safeguards that have to break down for cancer to develop. Imagine a healthy, normal cell just doing its job. The first step towards cancer is usually when this normal cell undergoes a mutation in its DNA. Now, a mutated cell isn't immediately a tumor. Ideally, one of two things should happen.
Right, the body has defenses.
Exactly. Either the cell successfully repairs the DNA mutation, returning to its normal state, which happens all the time, actually, or if it can't repair the damage, it undergoes programmed cell death or apoptosis. It basically self-destructs, eliminating the faulty cell before it can cause harm.
Like a cellular cleanup crew, a self-destruct sequence.
That's a great way to think about it. But here's where things go wrong in cancer. Cancer develops when the mutated cell not only fails to repair its DNA, but also fails to undergo apoptosis. It dodges that self-destruct command. This allows the mutated cell to survive and, crucially, to proliferate uncontrollably. As it divides, it can learn tricks to escape the body's immune system, which would normally recognize and destroy abnormal cells.
So it becomes invisible to the guards.
In a way, yes. Once it escapes immunity, it gains the ability to invade surrounding tissues and eventually spread to distant sites like the brain, lungs, liver, bones, leading to metastases. It's a multi-step breakdown in fundamental cellular processes, multiple failures.
That makes it incredibly clear why cancer is such a formidable opponent. It's not just one thing going wrong. And speaking of fundamental processes, our sources dive deep into the genetic blueprint of cancer, categorizing the genes involved into 3 critical groups. Can you explain those key players for us?
Yes, absolutely. Genes associated with cancer generally fall into 3 vital categories, each playing a different role in cell control. Think of it like managing a car. First, we have genes that induce cellular proliferation. Think of these as the gas pedals for cell growth.
Accelerators.
Exactly. Examples include growth factors like FGF and IGF. They're signals telling cells to grow. Then there are growth factor receptors like enzyme or ERPB, which are the on switches on the cell surface that receive those signals. And finally, internal messengers called signal transducers like SERAC or RAS relay the grow command inside the cell, and transcription factors like MY or FOS then turn on the specific genes that actually make the cell divide.
So when these gas pedals get stuck on go, you get uncontrolled growth. Makes sense.
Precisely. The second category is arguably just as critical: genes that suppress tumors and inhibit cell proliferation. We often call these tumor suppressor genes, and they are essentially the brakes of the cell cycle.
The brakes, okay.
Key examples include ARB, which suppresses retinoblastoma, P53, often called the guardian of the genome because it's so central in stopping tumor growth and initiating DNA repair, and APC, a suppressor involved in colon polyps. Their normal job is to halt abnormal cell growth or trigger repair or even apoptosis if things are too damaged.
And if those brakes fail, the cell just keeps dividing without restraint.
That's right. It can't stop itself. Finally, the third category involves genes that regulate programmed cell death or apoptosis. In a healthy cell, if a cell is damaged beyond repair, apoptosis ensures it's eliminated safely. A significant example here is BCL2, which actually suppresses this natural cell death.
Wait, it suppresses cell death, so it keeps cells alive.
Yes, its normal role is complex, but in the context of cancer, if a gene like BCL2 is overactive, it can prevent these damaged, potentially cancerous cells from dying when they should. It allows them to persist and accumulate more mutations.
Okay, I see. So during carcinogenesis, these vital genes, the gas pedals, the brakes, the self-destruct regulators, become dysregulated. What does that actually mean, and what's the result?
Dysregulation means their normal function is severely disrupted. This usually happens because of genetic alterations like a gene being deleted entirely or mutated so the protein doesn't work, or maybe its promoter is silent so the gene isn't even turned on. And the result is precisely what we discussed earlier: uncontrolled cell proliferation and the evasion of apoptosis. This creates the perfect storm for cancer development. What's really crucial to understand, though, is that cancer rarely involves just a single faulty gene. It typically involves mutations in more than one gene.
Ah, right. Multiple hits.
Exactly. It's often a complex interplay of several messed up genes, which makes single-target treatments incredibly challenging. And this complexity highlights why a nuanced approach like gene therapy is so promising.
Right. That complexity, then, is exactly why gene therapy for cancer has generated such intense interest. It sounds like it offers a truly targeted and sophisticated approach compared to, say, traditional chemo that hits everything.
It absolutely does. Gene therapy is emerging as a powerful approach because it allows for the precise targeting of specific genes within cancer cells, or sometimes even in the surrounding healthy tissue, to help fight the cancer. This makes it applicable for both diagnosing malignancies, potentially, and more excitingly, for treating them. The sheer volume of research underscores this interest. Our sources indicate that over 1,300 gene therapy trials have been published or are currently in progress.
Wow.
And more than 780 of those specifically directed against cancer. That's a massive investment of scientific effort and resources.
That's an astonishing number of trials. It really highlights the investment and, well, the hope in this field.
It truly does. And because of cancer's inherently complex nature, these gene therapy strategies are also incredibly intricate and highly specific. They're often tailored to different cancer types, different stages, different genetic profiles. It's not a single magic bullet, but rather a sophisticated toolkit where each tool is designed for a very particular job.
Okay, let's dive into some of those diverse strategies now. One of the most intuitive sounds like leveraging the body's own defense system: immunomodulation. How does gene therapy help unleash our body's own army against cancer?
Right. Immunomodulation is a strategy designed to essentially supercharge the human immune system's inherent ability to recognize and mount effective responses against tumor antigens, those flags on cancer cells. The challenge, though, is that cancer cells are incredibly cunning. They often employ various mechanisms to hide from or suppress the immune system. They evade detection.
I put on camouflage.
Exactly. Gene therapy aims to overcome these evasion tactics, essentially ripping off the camouflage and giving the immune system a clearer picture of the enemy, or maybe even giving the immune system better weapons.
So we're giving the immune system a better map and better gear.
That's a good analogy. We can do this by targeting various cell types. We could target the tumor cells themselves, or surrounding host cells, or critical immune cells like T cells or antigen-presenting cells, APCs, such as dendritic cells. T lymphocytes, for example, are a particularly attractive target for genetic modification because they are central players in our adaptive immunity, the part that learns to fight specific threats.
How do they actually deliver these genetic modifications into the cells? What are the methods?
Gene delivery can be done in various ways. In vivo, where the gene therapy vector is delivered directly into the patient's body, maybe injected into the tumor or intravenously. In vitro, meaning in glass, basically in a lab dish. Or ex vivo, which is quite common for immune cell therapies. Here, cells like T cells are removed from the body, genetically modified in the lab, expanded to large numbers, and then infused back into the patient.
Ex vivo sounds quite involved, but potentially very controlled.
It is. And the transgenes, the genes we introduce, can code for different things. They might code for cytokines. These are signaling molecules that act like alert signals or battle cries to recruit and activate other immune cells. Or they could code for co-stimulatory molecules, which are like a second signal needed to fully activate immune cells to fight effectively. Or even tumor-associated antigens themselves to sort of vaccinate the immune system against the cancer.
Could you give us some specific examples of how this immunomodulation plays out in practice? What do these strategies look like?
Certainly, some early clinical protocols, for instance, involve taking tumor-infiltrating T lymphocytes, or TILs. These are T cells that have naturally migrated into a tumor, showing they recognize something there. Researchers would extract these TILs, expand them in the lab, and enhance their activity by introducing cytokine genes before reinfusing them.
Boosting the natural warriors.
Exactly. We can also genetically modify T lymphocytes ex vivo by transferring cytokine genes, or even more precisely, by transferring genes for tumor antigen-specific T cell receptors, TCRs. Or perhaps even more famous now, chimeric antigen receptors, or CARs. These CARs are engineered receptors that have an extracellular domain designed to specifically bind to a tumor antigen and an intracellular domain designed to strongly activate the T cell upon binding.
CAR-T therapy. We hear a lot about that. It sounds like giving T cells a highly specialized targeting system and a turbo boost.
That's precisely what it is. Other strategies include inducing a strong local inflammatory response, for example, by injecting tumor cells or even harmless fibroblasts that have been genetically modified to continuously secrete immune-activating cytokines right at the tumor site, create a hotspot for the immune system. We can also try to force tumor cells to upregulate molecules like MHC class I or I and co-stimulatory molecules on their surface. Cancer cells often downregulate these to become invisible to T cells, so restoring their expression helps the immune system see them again.
And what about finding new targets? Are we still discovering what the immune system could potentially attack on cancer cells?
Oh, absolutely. That's a huge area of research. We can deliver tumor-derived antigens as naked DNA or via viral vectors, effectively presenting them to the immune system in a way that hopefully generates a stronger, more specific response like a therapeutic cancer vaccine. And advanced methods like SEREX, which is serological analysis of tumor antigens by recombinant expression cloning and microarray techniques, are constantly being used to discover novel tumor antigens that could be future targets. Beyond gene delivery into cells, antibody-based immunotherapy is also a powerful tool within this broader immunomodulation strategy. Here, scientists develop monoclonal antibodies designed to specifically target tumor antigens, either killing the cells directly or flagging them for immune destruction.
It's fascinating how many different angles there are to just this one strategy, the sheer ingenuity in boosting the immune system. But what if, instead of just enhancing the body's natural defenses, we could turn cancer's own processes against it? That brings us to what sounds like a pretty dramatic, but maybe highly effective tactic, the suicide strategy in gene therapy.
It does sound dramatic, doesn't it? And it's quite ingenious. The suicide strategy, more formally known as pre-drug converting enzyme therapy, cleverly combines classical chemotherapy with gene transfer technology. The core concept is to precisely localize the cytotoxic action of a drug directly to the tumor, thereby minimizing the devastating side effects on healthy neighboring cells.
So you're not just blasting the whole body with chemotherapy, you're making the cancer cells themselves activate the poison, turning them into their own little poison factories.
Precisely. That's a great way to put it. Here's the basic mechanism. First, a copy of the gene or cDNA for a specific prodrug converting enzyme is delivered specifically into the tumor cells, usually using a viral vector or some other delivery system. Then, the patient is given a prodrug, which is a compound that is non-toxic, or at least much less toxic in its initial form. Once this non-toxic prodrug reaches the tumor cells that are now expressing this new enzyme, the enzyme converts the prodrug into its active, highly cytotoxic form. This means the drug's lethal effects are largely restricted to the tumor area where the enzyme is being made.
Okay, that makes immediate sense. A huge improvement over traditional chemotherapy, which is notorious for its systemic toxicity and collateral damage to healthy tissues.
Absolutely, and what makes the strategy even more powerful is something called the bystander effect. This is a really fascinating phenomenon. It significantly enhances the efficacy because it leads to the killing of even uninfected tumor cells that are nearby the ones that actually received the gene.
How does that work? How do the cells that didn't get the gene die?
It can happen through several mechanisms. The activated toxic drug metabolites might pass directly from the infected cell to its uninfected neighbors through tiny channels called gap junctions, or these toxic substances might be released in small vesicles that get taken up by nearby cells. And interestingly, the dying tumor cells can also trigger local inflammation and anti-tumor immune responses that help kill off other tumor cells. So a relatively small number of initially transduced cells can have a widespread destructive effect within the tumor, like setting off a chain reaction.
That is incredible. It means you don't necessarily have to perfectly hit every single tumor cell with the gene delivery for the treatment to be effective. Can you give us a couple of prominent examples of these prodrug systems?
Yes. Two systems have been widely studied and illustrate this well. One uses the herpes simplex virus thymidine kinase gene, often abbreviated HSV-TK, along with the prodrug ganciclovir, or GCV. The HSV-TK enzyme phosphorylates GCV, converting it into a form that gets incorporated into DNA during replication and stops it, essentially killing dividing cells. Another common system uses the enzyme cytosine deaminase, or CD, which converts the relatively non-toxic prodrug 5-fluorocytosine 5-FC into the highly toxic chemotherapy agent 5-fluorosyl 5-FU. 5-FU messes up both DNA and RNA synthesis. Both of these systems show a significant bystander effect, which is key to their potential effectiveness.
So different enzymes, different prodrugs hitting different cellular processes, but the same fundamental principle of localized activation and bystander killing. And how are these suicide genes actually delivered to the tumor cells in the first place?
Delivery methods vary widely and are constantly being refined. They include non-viral methods like using liposomes or polymers to package the DNA, as well as various viral vectors, retroviruses, adenoviruses, and even specifically engineered oncolytic viruses that preferentially replicate in cancer cells can be used to deliver these enzyme genes. Studies have shown that, thanks to that bystander effect, sometimes transducing as little as 10% of the tumor mass can potentially lead to complete tumor eradication in experimental models.
That sounds incredibly promising, almost too good to be true.
Yeah.
What are the limitations? There must be challenges.
Indeed. As with many gene therapy strategies, one major limitation is the overall efficiency of in vivo gene transfer. Getting the gene into enough tumor cells in a living patient is tough. Even with direct intratumoral injection, only a fraction of tumor cells might actually get transduced, and the distribution within the tumor can be uneven. And as we discussed, the bystander effect, while a massive boon for killing untransfected tumor cells, also carries a potential risk. If the effect spreads too far, or if the enzyme gets expressed in some normal cells, it could lead to toxicity in healthy tissues. It's a delicate balance to strike.
Right, a potential double-edged sword. So how are researchers trying to overcome these hurdles? What's the next step?
There are several active areas of research. Improving vector distribution within the tumor is key. This might involve exploring different delivery routes, like delivering the therapy directly into specific body cavities, say intravesical for bladder cancer or intraperitoneal for ovarian cancer, or using techniques like bulk flow administration to push the vector further into the tumor mass. Another strategy is employing replication-competent vectors, particularly oncolytic viruses, which can naturally spread the therapeutic gene throughout the tumor as they replicate and seize cells. And finally, researchers are working on ways to specifically enhance the bystander effect, perhaps by increasing the number of gap junctions between tumor cells to facilitate drug transfer, or by developing vectors that target specific receptors highly expressed on cancer cells for more precise delivery.
This level of detail and targeting is just remarkable. Let's shift gears now to the strategy of using tumor suppressor genes and anti-oncogenes to restore balance in these rogue cells. It sounds like we're trying to put the brakes back on, or maybe fix the accelerator.
That's a perfect analogy. This strategy fundamentally involves counteracting the genetic dysregulation that drives cancer, the problems with those gas pedals and brakes we talked about earlier. It works in two main ways. Either by inactivating oncogenes that are overexpressed and driving growth, perhaps using tools like antisense molecules that block the oncogene's message, or dominant negative mutants that interfere with the oncogene's protein function. Or the other approach is to reintroduce functional copies of tumor suppressor genes that have been lost or mutated in the cancer cells, putting the brakes back in.
So it's about turning off the bad guys or bringing back the good guys, the genes that normally keep cell growth in check.
Exactly. Now, the primary goal here isn't necessarily a complete reversal of the tumor cells back to a perfectly normal state. That's often extremely difficult, if not impossible, given the multiple mutations usually involved in established cancers. Instead, the principle is often to identify weak points in the cell's now unbalanced regulatory network. The aim is to exploit these weaknesses to push the cancer cell towards either arresting its cell cycle stopping division or inducing apoptosis, that programs cell death. A deep understanding of the intricate pathways controlling signal transduction, cell cycle checkpoints, and apoptosis is absolutely critical for designing these strategies effectively.
Which specific genes are particularly promising in this area for putting those brakes back on or interfering with the accelerators?
Among the many tumor suppressor genes identified, p53 has really stood out and shown significant promise in early clinical investigations. It's such a central player in cellular control. Other important genes include caspases, which execute the apoptosis program, PATUN, another key tumor suppressor often lost in cancers, and BRCA1, famous for its role in breast and ovarian cancer susceptibility. These are all involved in regulating cell growth, repair, and death. We can also prevent cells from progressing through the cell cycle and dividing by introducing inhibitors of key cell cycle engines, like the proteins P16 and P21, which block cyclin-dependent kinases. What's potentially even more effective is combining these approaches. For instance, studies have shown an enhanced tumor-killing effect when you combine the reintroduction of multiple tumor suppressor genes, such as delivering both P53 and P16 together. It's like hitting the brakes and pulling the emergency brake simultaneously.
A coordinated attack, recognizing that cancer usually has multiple things wrong with it.
Precisely. This multi-pronged approach is often necessary because cancer itself is typically driven by multiple genetic hits. Now, when we compare this type of gene therapy to, say, small molecule inhibitor drugs like Gleevec, which targets a specific enzyme, gene therapy potentially offers broader capabilities. While small molecules mainly target enzymes and receptors, gene therapy can inhibit oncogenes using things like transdominant anti-oncogenes, antisense oligonucleotides, or even the really powerful RNA-interfering nucleotides like siRNAs or hRNAs. This provides a wider array of targets and mechanisms to try and restore cellular balance.
But what are the challenges with this approach? You mentioned delivery before. Is that still the main issue here, especially if the patient's immune system might already be weakened by the cancer or previous treatments?
Yes, delivery remains the main hurdle. Achieving efficient, high-level gene transfer into a sufficient percentage of the tumor cells is critical for this strategy to work effectively in vivo. In situations where the patient's immune system might be compromised, you can't necessarily rely on it to help clear out remaining cancer cells. So the gene therapy itself really needs to do the heavy lifting, potentially requiring very high transduction efficiency to completely eradicate the tumor. With current vector technologies, achieving that consistently high efficiency throughout a solid tumor mass can still be challenging. The full potential of this highly tumor-specific method really depends heavily on continued advancements in vector development, making them better at penetrating tumors and delivering their payload effectively.
Our sources also provided a great table highlighting some key tumor suppressor genes and the cancers they're linked to when mutated. Could you maybe elaborate on one example like RB1? How does it act as a brake, and what happens when it's faulty?
Yes, the RB1 gene is a fantastic classic example of a tumor suppressor. It's located on chromosome 13, and it normally produces a protein, PRB, that acts as a crucial gatekeeper for the cell cycle. Specifically, PRB binds to another protein called E2F, which is a transcription factor. When TRB is bound to E2F, E2F cannot activate the genes needed for DNA replication and cell division. So PRB essentially holds the cell cycle in check at a critical point called the G1 checkpoint, preventing cells from entering the S phase where DNA is copied. It's a key break. However, if the RB1 gene is mutated or deleted, the resulting faulty or absent PRB protein fails to bind and inhibit E2F. This unleashed E2F is then free to turn on the genes that stimulate the cell cycle, leading to uncontrolled cell division and eventually tumor formation. This is famously seen in retinoblastoma, a cancer of the eye. There's a hereditary form where individuals inherit one faulty copy of RB1, making them highly susceptible to developing multiple tumors because only one more hit is needed in a retinal cell. And there's a non-hereditary form where two separate mutations have to occur in the RB1 gene in the same retinal cell later in life, which usually results in just a single tumor. It's a very clear illustration of how losing a cellular break can lead directly to cancer.
That really brings the concept of a tumor suppressor gene to life. Okay, now let's talk about something that sounds even more direct. Using viruses not just as delivery vehicles, but as targeted assassins of tumor cells. This is tumor lysis by recombinant viruses, or viral oncolysis.
Right, this is a truly fascinating strategy, and actually the idea isn't new. Observations of tumors shrinking after viral infections go back to the early 1900s. Adenoviruses were first explored somewhat crudely for cancer treatment as early as 1957, but the modern approach is much more sophisticated. The basic idea is to engineer recombinant viruses to act as selective anti-cancer agents. These viruses are designed to specifically infect and then literally lyse or burst open tumor cells while ideally leaving normal cells unharmed.
So the virus acts like a Trojan horse, getting inside the cancer cell, replicating like mad, and then destroying the cell from within.
Precisely. The process usually begins with delivering the virus, often by direct intratumoral injection, though systemic delivery is also being explored. The virus then transduces, or infects, the cancer cells. Crucially, these engineered viruses are designed to replicate preferentially within these cancer cells. This replication leads to the production of many new viral particles and ultimately causes the lysis, the bursting of the infected cell. This releases the new viral progeny. These newly released viruses then infect neighboring cancer cells, spreading the infection and continuing the cycle of replication and cell lysis throughout the tumor. The potential for spread and amplification within the tumor means the transfer efficiency in this case can be conceptually very high, which is a major advantage over non-replicating vectors.
That sounds incredibly powerful, but also carries an obvious risk, doesn't it? How do they ensure the virus only targets the tumor and doesn't start replicating uncontrollably in healthy tissue? Containment must be key.
Absolutely. That's the major challenge and the focus of intense engineering efforts. Researchers have developed several clever conditional replication strategies to try and restrict viral replication specifically to the tumor site. One well-known method relies on the status of the p53 tumor suppressor gene, which we know is mutated or lost in about half of all human cancers. Adenoviruses, for example, can be engineered by deleting a viral gene called E1b55kD. This modified virus can replicate effectively in tumor cells that lack functional p53. However, in normal cells that do possess functional p53, the infection by this modified virus actually triggers apoptosis, effectively disarming the virus in healthy tissue.
Wow, that's incredibly clever. So the virus essentially weaponizes a common defect found in cancer cells against them. That's quite elegant.
It is, isn't it? Similarly, another approach involves modifying a different adenoviral gene, E1A, so that the virus can only replicate efficiently in tumor cells that have defects in their PRB pathway, another common feature of cancer cells linked to that RB1 gene we discussed. Yet another elegant approach involves placing the essential viral replication genes, like E1A, under the control of tissue-specific or cancer-specific promoters. For example, using the probasin promoter restricts replication largely to prostate cells, or using the MUC1 promoter targets certain breast cancers. This means the virus's replication machinery is only switched on when it finds itself inside a cell expressing these specific promoters, which are typically much more active in cancer cells than in normal tissues. It literally turns the virus into a smart bomb designed to detonate only at the correct cellular address.
These conditional replication strategies are amazing. Are there other types of viruses being explored for this oncolytic purpose besides adenoviruses?
Oh yes, definitely. Beyond adenoviruses, herpes simplex virus, or HSV, the virus that causes cold sores, is also being extensively modified for oncolytic therapy, particularly for brain tumors due to its natural tropism for nerve cells. Researchers have created HSV strains, for example, by deleting the viral thymidine kinase gene, HSV-TK. This makes the virus dependent on the cellular version of the enzyme, which is much more active in rapidly dividing cancer cells than in non-dividing normal brain cells. So the virus replicates well in the tumor, but poorly in the surrounding healthy brain tissue. There are first and second generation oncolytic HSV viruses, with the later generations often incorporating additional mutations to further enhance tumor-specific replication and reduce any remaining potential for neurovirulence, making them safer and potentially more effective.
And what about something less common, like Newcastle disease virus? And I read that was also being looked at.
That's right. Newcastle disease virus, or NDV, is another interesting candidate. It's an avian virus that causes significant disease in birds, but generally causes little or no illness in humans. Yet it has shown potent cytotoxic effects on a wide range of human tumor cell lines in the lab, while often sparing normal human cells. Its exact mechanism of tumor specificity isn't fully understood, but might involve cancer cells having a defective interferon response, a key antiviral defense. Or perhaps NDV-inducing tumor necrosis factor, TNF, mediated killing specifically in tumor cells. The future of this strategy is very promising, as our increasing knowledge of both tumor biology and virology allows for the rational design and generation of even more sophisticated and tumor-selective recombinant oncolytic viruses.
It's a rapidly evolving field. What factors ultimately determine how efficient these viral assassins will be in a real patient?
Several factors come into play and influence their overall efficiency. These include the specifics of the viral genome itself, its size, what genes it carries, the eclipse period, the time it takes for the virus to replicate inside a cell before bursting it, matters for the speed of spread. The number of infectious new variants produced per localized cell is also critical for propagation. And, importantly, the host's own immune response is a major factor. The immune system might recognize the virus as foreign and try to clear it before it can eliminate the tumor, so managing or modulating the immune response is often part of the strategy. Finally, the characteristics of the tumor itself, like how well vascularized it is or how much dense fibrous tissue it contains, can impact the virus's ability to penetrate and spread effectively. It's a complex interplay between the virus, the tumor microenvironment, and the patient's immune system.
It's all about precision and overcoming biological barriers, isn't it? Which brings us to another really unique approach: anti-angiogenic and anti-proteolytic gene therapy. This sounds completely different, like it's targeting the tumor support system, literally starving it or preventing it from breaking out.
Exactly. This strategy takes a different tack. Instead of directly killing the cancer cells, it targets two crucial processes that tumors rely on to grow and spread. The first is angiogenesis, which is the formation of new blood vessels. Tumors desperately need these new vessels to supply them with nutrients and oxygen to grow beyond a tiny size. The second is proteolysis, which is the breakdown of the extracellular matrix, the scaffolding between cells. Tumors use enzymes called proteases to degrade this matrix, allowing them to invade surrounding tissues and metastasize. So this approach aims to cut off the tumor supply lines and block its escape routes.
What are the big advantages of targeting these processes compared to, say, directly targeting the cancer cells with a suicide gene or an oncolytic virus?
One of the major potential benefits is the extracellular action of the gene products. The therapeutic molecules produced anti-angiogenic factors, or protease inhibitors, work outside the cells. This means you don't necessarily need to transduce every single tumor cell. In fact, you could even target normal, healthy tissue near the tumor to become a factory producing these inhibitors, which can then diffuse and act on the tumor microenvironment. This approach might also be well suited for long-term treatments or even preventing recurrence, perhaps aiming for sustained low levels of these inhibitors. And potentially, these secreted proteins might be less immunogenic than viral vectors or proteins expressed inside cells.
So by starving the tumor of its blood supply with anti-angiogenesis, you could potentially avoid the tumor developing resistance, which happens so often with drugs that target the cancer cells themselves.
That's one of the key hopes, yes. Angiogenesis targeting is incredibly promising because the endothelial cells lining the blood vessels are genetically stable normal cells, unlike the rapidly mutating cancer cells. This should make it much harder for the tumor to develop resistance. It's also a potentially broad approach, as most solid tumors rely on angiogenesis. And critically, the target cells, the blood vessels, are generally easily accessible via the bloodstream for systemically delivered therapies. Examples of anti-angiogenic genes that have been delivered using gene therapy vectors include endostatin and angiostatin, which are natural inhibitors of angiogenesis, certain cytokines like IL-12, which have anti-angiogenic effects, or soluble forms of VEGF receptors that act as decoys to soak up the tumor's pro-angiogenic signals.
And are there advantages to delivering the genes for these inhibitors rather than just injecting the inhibitor proteins themselves, which is also done?
Definitely. Gene therapy, or molecular therapy as it's sometimes called in this context, offers several potential advantages over just giving repeated injections of the protein. By using vectors, perhaps targeted vectors, you might achieve higher and more sustained concentrations of the anti-angiogenic factors directly within the tumor microenvironment. This could increase efficacy and potentially reduce systemic side effects that might occur with high circulating levels of the protein, which could interfere with normal processes like wound healing. It could also be lower cost in the long run compared to manufacturing and repeatedly administering large amounts of recombinant protein. And crucially, gene therapy might provide a more constant, steady presence of the inhibitor, which could be more effective than the peaks and troughs seen with protein injections. This tumor-static nature, slowing or stopping growth rather than immediate killing, makes anti-angiogenic therapy potentially very important in settings like before surgery to shrink a tumor or after surgery to prevent residual cells from growing or metastasizing.
That makes a lot of sense. And then there's the other side of this coin. Antiproteolytic therapy, stopping the tumor from breaking down the matrix and spreading.
Right. As we said, cancer progression, particularly invasion and metastasis, involves significant degradation and remodeling of the extracellular matrix, the ECM. This is largely carried out by various classes of proteases, enzymes like matrix metalloproteinases, MMPs, serine proteinases, and others. The strategy here involves tipping the balance back by increasing the levels of the natural inhibitors of these proteases. Key inhibitors include plasmin activator inhibitors, or PAI, and the family of tissue inhibitors of metalloproteinases, or TIMPs. PAI, for instance, has been quite extensively evaluated in preclinical models for its anti-tumor efficacy.
Have there been successful experiments showing that boosting these inhibitors with gene therapy can actually stop tumor spread?
Yes, there have been promising preclinical results. For example, experiments in rats using adenovirus vectors to deliver TIMP2 or TIMP3 genes have shown significant reductions in primary tumor growth, sometimes by 60–80%, and even more dramatically, reductions in lung metastases by up to 90% in some models. There's even this fascinating impregnation approach, where the genes for these inhibitors are transferred into normal tissue surrounding a tumor, essentially creating a defensive barrier that resists invasion. This has also shown significant tumor growth reduction in animal models. However, it's also a complex biological system. Some studies have yielded contradictory results. For instance, the role of PAI seems complex, with some studies suggesting it promotes invasion in certain contexts. This highlights that we still need a deeper understanding of the intricate interplay between specific proteases and inhibitors in different tumor types and stages to really optimize this therapeutic strategy. It's an active and complex area of research.
It sounds like a lot of fine-tuning is still needed there. Okay, moving on to something that sounds almost counterintuitive at first, using drug resistance genes. Why would we ever want to make cells resistant to cancer drugs?
It does sound paradoxical, doesn't it? But this strategy is actually incredibly clever, and it's not aimed at the cancer cells at all. The goal here is to make the patient's normal, healthy cells resistant to the toxic side effects of chemotherapy. Instead of directly killing tumor cells, this approach focuses on protecting vulnerable, healthy tissues, particularly the hematopoietic precursor cells in the bone marrow. These cells divide rapidly and are very sensitive to chemotherapy, leading to common side effects like low blood counts, infection risk, and fatigue. These side effects often limit the dose of chemotherapy that can be safely given.
Ah, I see. So it's about protecting the patient's healthy cells, specifically the bone marrow stem cells, so that they can tolerate higher, more effective doses of chemotherapy directed at the cancer. It's enabling a stronger attack on the tumor.
Exactly. You shield the good guys so you can hit the bad guys harder. The mechanism involves delivering drug resistance genes into these healthy hematopoietic stem cells, usually ex vivo. A classic example is the multiple drug resistance gene 1, or MDR1, which encodes a protein called P-glycoprotein. This protein acts like a pump on the cell surface, actively effluxing or pumping out a wide range of common chemotherapy drugs from the cell, thus protecting it. Other potential genes encode enzymes that can detoxify chemotherapy agents inside the cell, like glutathione S transferases or O6-alkylguanine transferase.
And what's the progress been like in this area? Has it moved beyond just theory in animal models?
There's been significant progress, though it's been challenging. Early clinical trials often struggled with low efficiency of gene transfer into the human stem cells and low levels of expression of the resistance genes, meaning the protective effect wasn't always strong enough. However, more recent reports using improved vectors and transduction techniques have demonstrated better success, showing long-term persistence of the gene-modified cells and evidence of hematopoietic recovery in vivo after chemotherapy. Perhaps the most compelling evidence that we can successfully transduce human hematopoietic stem cells for long-term benefit comes from the gene therapy trials for X-linked severe combined immunodeficiency, or SCID. While that's treating a different disease, it proved the principle. In those trials, introducing a correct copy of the faulty gene into patients' stem cells has led to immune system recovery in the vast majority of treated children, showing these modified stem cells can engraft and function long-term. This success offers immense hope and learning for applying similar techniques to confer drug resistance. Further clinical improvements are expected with more efficient transduction methods and perhaps optimized drug selection strategies to really leverage the resistance.
That's a huge step toward making chemotherapy potentially more tolerable and more effective. It's a really smart, indirect approach. Okay, finally, before we transition to our next major topic, what are marker genes used for in gene therapy? They sound like they're not directly therapeutic.
That's right. Marker genes are essentially tracking devices. They aren't intended to treat the disease themselves, but are used for tracing purposes. They account for about 6% of all gene therapy clinical trial protocols, according to our sources. They have enabled some important practical applications. For instance, they've been used to visualize and quantify any residual tumor cell contamination in bone marrow harvested from a patient for autologous transplantation. Marking the cancer cells allows detection if they inadvertently get reinfused, which is crucial for preventing relapse. They are also very valuable in research settings to trace the long-term survival, distribution, and fate of genetically modified cells, whether they be hematopoietic stem cells or engineered T cells after they've been put back into the body. They allow researchers to literally follow the cells and see where they go and how long they last.
Okay, that clarifies their role. Let's unpack this next section because it really gets into the nitty-gritty of precise genetic editing. Chimeroplasty. How does this technique allow for, well, rewriting the genetic code, potentially base by base?
Chimeroplasty is a fascinating and somewhat distinct non-viral technique specifically designed for targeted gene correction at a very fine level. Imagine having a tiny, super precise search and replace tool for your DNA. It involves synthetically creating a hybrid molecule, a chimera that consists of both RNA and DNA segments carefully stitched together. This hybrid molecule is then designed to find a specific spot in the genome and repair very small genetic alterations, such as single point mutations or perhaps tiny deletions or insertions of just a few base pairs. And what's quite remarkable is that this general approach has been explored in both plant and animal cells.
Where did this ingenious idea come from? Sounds different from just adding a whole new gene.
The concept is largely credited to Dr. Eric Pamiuk. He observed back in the 90s that the rate of genetic recombination DNA strands exchanging pieces seemed to be enhanced during the process of transcription when a gene is being read out into RNA. This led him to the realization that perhaps by coupling RNA with DNA into a single molecule, he could increase the stability of the molecule inside a cell and also leverage the cell's natural recombination and repair pathways. The idea was that this RNA-DNA hybrid could target a specific gene sequence and trigger the cell's own machinery to make a precise correction using the chimera as a template. His early work, and that of others, explored correcting known oncogenic mutations, like in the RAS gene and other point mutations, even showing some success in models of diseases like sickle cell anemia.
So it's literally trying to trick the cell into fixing its own typo, using this hybrid molecule as the correct spelling guide. What does this paperclip-shaped structure, as the sources call it, actually look like, and how is it designed for stability?
Yes, spelling guide, a good analogy. The chimeroplast itself is often described as having a distinct paperclip-shaped structure. It typically consists of a double-stranded DNA segment, which contains the correct sequence information, flanked on the ends by short strands of RNA. This unique hairpin-like design is thought to be crucial for its stability and function.
And what are the protective elements? Cells are pretty good at degrading foreign bits of DNA or RNA, so how does this chimera survive long enough to work?
Right, protecting it from degradation is key. The chimeraplast design incorporates several clever protective features. First, it often includes a special cap structure at the very ends, sometimes referred to as a hairpin cap. This acts like a tiny shield, guarding the RNA-DNA chimera from being chewed up by the cell's natural cleanup enzymes, called exonucleases, and also potentially resisting unwinding by cellular helicases. Second, parts of the RNA backbone are chemically modified, typically through methylation of the ribose sugar at the 2OH position. This modification acts like weatherproofing, protecting the RNA component from being attacked and cleaved by another cellular enzyme called RNase H, which specifically degrades the RNA part of RNA-DNA hybrids. Finally, the short RNA strands themselves are thought to play an active role, not just in stability, but potentially in activating the oligonucleotide for homologous recombination, helping to guide the chimera to the correct target site in the DNA and initiate the repair process.
That's some incredibly intricate molecular engineering. How do they design the DNA part of the chimeraplast to specifically target just one mutation in the vast expanse of the genome?
The design process has to be very precise. First, scientists synthesize a short stretch of DNA sequence that exactly matches the sequence of the target gene surrounding the mutation they want to correct. This homologous sequence is what allows the chimera to find and bind to the right spot in the genome. The crucial second step is to build the correct base into the synthetic DNA sequence at the exact position corresponding to the mutation in the patient's gene. So when the chimeraplast finds its target and binds, this creates a very specific mismatch. The chimera has the right base, the gene has the wrong base right at the site of the mutation. It's this mismatch that is recognized by the cell's repair system.
And how does the cell's own repair system then edit the DNA using the chimera as the accurate blueprint? What happens next?
When the chimeraplast binds to the target DNA sequence, that single mismatched base pair acts like a red flag, signaling to the cell's own endogenous repair machinery that something is wrong. The cell then activates its mismatch repair system, the same system it uses to fix errors made during normal DNA replication. This machinery recognizes the mismatch and typically excises the incorrect base from the genomic DNA strand. It then uses the sequence on the bound chimeraplast, which carries the correct base as the template, to synthesize the repair patch, effectively correcting the mutation in the gene. Once the target gene is successfully repaired, the chimeraplast itself is no longer needed and is naturally degraded by the cell, leaving behind only the corrected target DNA sequence.
And how do these chimeraplasts actually get into the cells and then into the nucleus where the DNA is?
Delivery is, as always, a challenge. Various methods have been explored for getting these chimeric molecules into cells or tissues. These include binding them to specific ligands that recognize receptors on target cells, like liver cells, encapsulating them in protective lipid nanoparticles, liposomes, complexing them with synthetic polymers, or in plants, even coating microscopic gold particles with the chimeraplasts and literally shooting them into the cells using a gene gun. Once inside the cell cytoplasm, the chimeraplasts need to make their way into the nucleus, find and bind to their specific target DNA sequence, and then successfully invoke that mismatch repair mechanism.
Okay, so assuming it gets there and works, what are the most promising potential applications of chimeraplasty given this precise find and replace mechanism?
Chimeraplasty, in theory, holds immense potential, especially for gene therapy aimed at correcting specific point mutations. Diseases caused by a single letter change in the DNA code, like sickle cell anemia or, as we'll discuss, Crigler-Najjar syndrome, seem like ideal candidates. Chimeraplasty is generally thought to be most efficient at altering just one or maybe up to 3 consecutive base pairs in a gene. Another potentially very significant role is in gene therapy for autosomal dominant mutations. Remember, in these diseases, having just one mutated copy of the gene is enough to cause the disorder. Traditional gene therapy, which usually just adds a normal copy of the gene, often can't overcome the negative effects of the faulty dominant protein being produced from the mutated gene. Chimeraplasty, however, offers the prospect of directly correcting the defective dominant gene itself, which could be a much more effective approach for these conditions.
That's a key difference. And it sounds like it could be a powerful tool for research, too, beyond just therapy, for understanding gene function.
Absolutely. Chimeraplasty could potentially be used to deliberately knock out or inactivate genes in specific cells or tissues. By introducing a small change that disrupts the gene's function, scientists could study the effects of losing that gene. This could be used to create cellular or even animal models, knockout models, for various human diseases, helping us understand the disease process and test potential therapies. It offers a way to manipulate gene function with high precision.
Those are some truly incredible potential applications. But technology like this rarely comes without hurdles. What are the current limitations or drawbacks that researchers are still trying to overcome with chimeroplasty?
Despite its elegance and promise, chimeroplasty faces several significant limitations that have, so far, prevented its widespread clinical application. First, as mentioned, it's really only suitable for making very small corrections, typically thought to be a maximum of 3 consecutive base pairs. It can't fix large deletions or insertions. Second, and perhaps the biggest problem, has been the unpredictable and often low rate of actual gene correction. Reported efficiencies have varied wildly in different studies and different systems, sometimes ranging from less than 1% up to maybe 40% in optimal conditions. The reasons for this variability and often low efficiency are still not fully understood, but likely involve issues with delivery, stability, and perhaps how effectively the cell's repair machinery engages with the chimera.
So it's not consistently effective yet, making it hard to rely on clinically?
Not consistently, no. Another potential concern is the risk of nonspecific or off-target alterations. Because the chimera relies on sequence homology to find its target, there's a risk that if there are other genes in the genome with very similar sequences, the chimera might bind there accidentally and introduce unwanted mutations, potentially causing harmful side effects. Specificity is crucial. Finally, the RNA-DNA hybrid molecule itself is inherently less stable than pure DNA. Studies suggest it typically degrades within the cell in about 48 hours or less. This gives the cell's repair mechanism a limited window of time to recognize the mismatch and perform the correction. These limitations collectively highlight why, despite the initial excitement, the efficiency and safety of chimeroplasty for therapeutic use in humans are still areas of active research and require significant improvement.
Okay, that puts it into perspective. Given those limitations, but also the specific potential for point mutations, let's turn our attention now to a disease that seems like a textbook candidate for this kind of precise correction. Crigler-Najjar syndrome. What is this disease and how does it manifest?
Crigler-Najjar syndrome is a rare inherited metabolic disorder. It's an autosomal recessive genetic disease, meaning a person needs to inherit 2 faulty copies of the responsible gene. Its defining characteristic is severe chronic hyperbilirubinemia, which basically means persistently high levels of a substance called unconjugated bilirubin accumulating in the blood. To understand why this happens, we need a quick look at how the body normally handles bilirubin. Bilirubin is a yellowish waste product formed mainly from the breakdown of heme, a component of old red blood cells.
Okay, so it's a natural waste product. What happens to it normally?
Normally, this initial form, unconjugated bilirubin, is fat-soluble, not water-soluble. It binds to a protein called albumin in the blood and gets transported to the liver. In the liver, a crucial enzyme called UDP-glucuronidyltransferase 1A1, usually just abbreviated as UGT1A1, performs a vital step called conjugation. It attaches molecules of glucuronic acid to the bilirubin. This conjugation step makes the bilirubin water-soluble, allowing it to be excreted from the liver into the bile, then into the intestines, and finally eliminated from the body. In Crigler-Najjar syndrome, the UGT1A1 enzyme is either completely inactive or severely deficient due to mutations in the UGT1A1 gene, so this conjugation step can't happen properly.
And if it can't be conjugated and made water-soluble, the bilirubin just builds up. What's the danger there?
Exactly. The unconjugated bilirubin accumulates in the blood to very high levels. And because it's fat-soluble, if the levels get too high, the bilirubin that's not bound to albumin can cross the blood-brain barrier and deposit in the brain tissue. This leads to a devastating condition called kernicterus, or bilirubin encephalopathy, which causes irreversible neurological damage, things like cerebral palsy, hearing loss, developmental delays, and can often be fatal if not managed aggressively, especially in newborns.
So it's a defect in the liver's ability to process and detoxify this waste product, leading to potential brain damage.
Right.
Are there different levels of severity, different types of Crigler-Najjar?
Yes, there are 2 main types, based on the residual activity of the UGT1A1 enzyme. Type I is the most severe form. Here, there is a complete or near-complete absence of UGT1A1 enzyme activity. Patients have extremely high bilirubin levels, often above 20 mg/dL or 345 ml. This type is very difficult to treat, usually doesn't respond well to medication, and requires lifelong intensive management to prevent kernicterus. Type 2, sometimes called Arias syndrome, is generally less severe. Patients have severely reduced but still detectable UGT1A1 activity, maybe less than 10% of normal. Their bilirubin levels are typically lower than in type I, and they often show some response to treatment with drugs like phenobarbital, which can induce the remaining enzyme activity. Kernicterus is less common but still a risk.
What were the traditional treatments for this severe condition, especially type I, before gene therapy became a possibility? How did they manage these dangerously high bilirubin levels?
For type I Crigler-Najjar, the mainstay of treatment for decades has been intensive phototherapy. Patients, especially infants and children, have to spend 10-12 hours every single day, often while sleeping, under special high-intensity blue lights. This light exposure helps to convert the bilirubin accumulated in the skin into different, more water-soluble isomers that can be excreted without needing liver conjugation. It's life-saving but incredibly burdensome and becomes less effective as children grow older and their skin thickens, reducing light penetration. Other interventions, especially in newborns, might include exchange transfusions, essentially replacing the baby's blood to rapidly lower bilirubin levels, sometimes oral calcium salts or other agents used to try and bind bilirubin in the gut. Ultimately, however, for many type I patients, the only definitive cure and the only way to prevent eventual brain damage as phototherapy becomes insufficient is a liver transplantation. This is obviously a major surgical procedure with significant risks and requires lifelong immunosuppression.
Wow, daily hours under lights or a liver transplant? That really underscores the need for better options. It makes sense that gene therapy would be intensely explored. Our sources highlight the gun rat as a crucial animal model for this. What's the story behind the gun rat?
The gun rat is a really important part of the Crigler-Najjar story. It was discovered purely by chance back in 1934 by Charles Gunn at the University of Toronto. He noticed a mutant strain of Wistar rats that developed spontaneous jaundice, yellowing of the skin and eyes. It was later discovered that this jaundice was due to an autosomal recessive genetic defect that resulted in a lack of UGT1A1 enzyme activity, making it the perfect naturally occurring animal model, a direct analog of human Crigler-Najjar syndrome type I. At the molecular level, the defect in the gun rat UGT1A1 gene was eventually identified as a single-base pair deletion, causing a frame shift and a premature stop codon, leading to a non-functional enzyme.
So it's a living, breathing replica of the human disease, allowing researchers to test therapies directly relevant to patients. Why is it considered such an attractive model specifically for liver-directed gene therapy?
There are several compelling reasons why the gun rat has been invaluable. First, as we said, the disease is incredibly well defined, both biochemically, high unconjugated bilirubin, and molecularly, the specific gene defect is known. Scientists know exactly what they need to fix. Second, and this is really crucial from a therapeutic standpoint, studies showed that you don't need to restore 100% of normal UGT1A1 enzyme activity to have a major clinical benefit. Even restoring just 5–10% of normal activity is enough to significantly decrease serum bilirubin levels, often below the threshold considered dangerous for brain damage. This lowers the bar for therapeutic success. You don't need perfect correction. And third, the efficacy of any potential treatment is very easily monitored. You can simply take blood samples and measure the bilirubin levels, or even collect bile and measure the amount of conjugated bilirubin being excreted. This provides clear, quantitative, objective evidence of whether a therapy is working.
That makes it a fantastic proving ground. So let's explore the specific gene therapy approaches that have been tested in the gun rat for Crigler-Najjar, starting with plasmid-mediated gene therapy. How did that begin, and what were the initial findings?
The earliest attempts often involved using simple plasmids, circular pieces of DNA containing the correct human UGT1A1 gene sequence. Early investigations, like those by Wilk and colleagues mentioned in the sources, demonstrated proof of concept. They showed that if you could get this plasmid into isolated gun rat liver cells, hepatocytes, in a lab dish, in vitro, you could indeed restore their ability to conjugate bilirubin. So the gene itself worked.
But the challenge, as often happens in gene therapy, was translating that success from a petri dish into a living animal, right? Getting the plasmid into enough liver cells in vivo.
Exactly. Getting naked plasmid DNA efficiently into liver cells in a living rat proved much harder. When injected systemically, the efficiency was very low, largely because the plasmids were quickly degraded by enzymes within the liver cells, probably after being taken up into compartments called endosomes. To overcome this hurdle, researchers developed more sophisticated delivery strategies. One approach involved targeting the plasmid specifically to liver cells by attaching molecules that bind to the acylglycoprotein receptor, which is abundant on hepatocyte surfaces. They combine this targeted delivery with the administration of drugs like colchicine, which can help inhibit the degradation pathways inside the cell, allowing more plasmid DNA to reach the nucleus. This combined approach did lead to some success. A reported 25–35% decrease in serum bilirubin levels that lasted for several weeks in the gun rat. A definite improvement, but still transient.
Okay, a good step, but several weeks clearly isn't a lifelong cure. What about using chimeroplasty for Crigler-Najjar? You mentioned earlier it was suited for point mutations, and the gun rat has that single base deletion. Was it tried?
Yes, indeed. Given that the gunrat defect is a single base deletion, it seemed like a perfect target for chimeraplasty's proposed mechanism of precise gene correction. The idea was to design a chimeraplast that would align with the gunrat UGT1A1 gene sequence around the deletion site, but contain the correct sequence. The hope was that this would induce the cell's mismatch repair system to use the chimera as a template and essentially fix the deletion in the rat's own gene.
And what were the results reported from those experiments? Did it work?
There were reports, notably from Kren and colleagues, suggesting significant success. They administered chimeraplasts intravenously to gun rats, either mixed with a synthetic delivery polymer called PEI or encapsulated in liposomes. They reported achieving correction of the UGT1A1 gene in up to 20% of the liver cells. This was quite exciting at the time, as it suggested direct in situ gene repair was possible. However, it's really important to add a significant caveat here. As we discussed earlier regarding chimeraplasty's limitations, subsequent studies by many other research groups have raised serious doubts about the reproducibility and the actual efficiency of gene correction achieved via chimeraplasty in vivo. There is ongoing debate and investigation into the true efficacy of this approach, so its role in Crigler-Najjar therapy remains uncertain.
Right, the consistency issue. That brings us to using viruses as vectors, specifically adenovirus. What makes adenovirus potentially a strong contender for delivering the UGT1A1 gene to the liver in Crigler-Najjar syndrome?
Adenoviruses offer several key advantages that made them attractive early vectors for liver-directed gene therapy, including for Crigler-Najjar. First, when administered systemically, for example intravenously, adenoviral vectors naturally show a strong tendency to be taken up by the liver. They're quite efficient at infecting hepatocytes. Second, they are capable of mediating very high levels of gene expression shortly after infection, which is good if you need a lot of enzyme produced quickly. Third, adenoviral vectors can be produced at very high concentrations, or titers, making it feasible to administer large doses. And fourth, their genome is relatively large, allowing them to carry sizable foreign DNA inserts, like the full coding sequence for UGT1A1. Finally, a potential safety advantage is that the adenoviral genome typically remains episomal. It exists as a separate piece of DNA in the nucleus and doesn't integrate into the host cell's chromosomes. This avoids the risk of insertional mutagenesis, where integration could potentially disrupt other important genes and cause cancer, which is a concern with some other viral vectors like retroviruses.
Those sound like compelling advantages. But I recall you mentioning earlier that first-generation adenoviral vectors often cause strong immune responses. Was that a problem here too? Did the body fight off the treatment?
Yes, that turned out to be a major hurdle with the early first-generation adenoviral vectors used for Crigler-Najjar in the gunrat models. These early vectors typically had a key viral replication region, E1, deleted to prevent the virus from replicating. However, this didn't completely stop the expression of other adenoviral proteins from the vector genome. The expression of these residual viral proteins acted as foreign signals, triggering a quick and strong immune response from the host's T cells, which then attacked and eliminated the transduced liver cells. As a result, while these vectors initially worked well and reduced bilirubin levels significantly, the effect was transient. In gun rats, the therapeutic effect, the reduction in bilirubin, typically lasted only about 7 to 12 weeks before the immune system cleared out the gene-corrected cells.
So the cure didn't stick. How did researchers try to overcome that immune rejection problem to get longer-lasting effects?
Several strategies were explored to try and mitigate or evade this immune response. One approach involved temporarily suppressing the host's immune system around the time of vector administration, using immunosuppressive drugs like FK506 or cyclosporine. This could dampen the initial attack and allow for longer gene expression, especially if the vector needed to be readministered. Another strategy focused on modifying the vector itself. For instance, researchers tried reinserting certain adenoviral genes from the E3 region, which are known to naturally help adenovirus evade host immune responses. Alternatively, the vector could be engineered to co-express other immune-modulating proteins, like CTLA-4Ig, which can inhibit the co-stimulatory signals needed to fully activate T cells, essentially dampening the specific immune attack against the transduced cells.
That leads us logically to the next generation. Helper-dependent adenovirus, often called gutless vectors. This sounds like a major redesign specifically aimed at solving that immunogenicity problem. What makes them different and potentially much better?
This was indeed a major innovation, designed precisely to address the immunogenicity caused by viral protein expression from earlier vectors. The realization was that if viral proteins trigger the immune response, the best solution is to get rid of all the viral genes. Helper-dependent adenoviruses, or HD-ad vectors, achieve this. They lack all viral coding sequences. The only adenovirus components left are the short inverted terminal repeats, ITRs, at the ends, which are needed for DNA replication and packaging, and the packaging signal sequence itself. The entire rest of the viral genome, all the genes encoding viral proteins, has been deleted or gutted, hence the nickname gutless vectors. This space can then be filled with the therapeutic gene.
Wow, so absolutely no viral proteins produced at all. That must make a huge difference to the immune response.
Precisely. Because there's a complete absence of viral gene expression from the vector itself after it infects the cell, these gutless vectors are significantly less immunogenic and also less cytotoxic to the transduced cells compared to earlier generations. This translates directly into much more stable and longer-lasting expression of the therapeutic gene, like UGT1A1, in the liver cells in vivo. The results reported in gun rats using HD advectors encoding human UGT1A1 were quite remarkable. Administration of just a single optimal dose led to a profound, near-complete correction of the hyperbilirubinemia and 80% decrease in serum bilirubin levels that persisted for up to 2 years, which was essentially the lifespan of the study animals. This demonstrated the potential for very long-term, stable correction with this advanced vector platform.
2 years of efficacy from a single shot is incredible. That sounds like it's getting very close to a potential cure. Is there another vector that shows similar, or perhaps even better, long-term promise for Crigler-Najjar? What about AAV?
Yes, adeno-associated virus, or AAV, vectors have also emerged as highly favored vectors for Crigler-Najjar syndrome and indeed for many other genetic diseases targeting the liver. AAD has a number of inherent advantages that make it very attractive. First, wild-type AAV is not known to cause any human disease. It's considered non-pathogenic. The vectors used are replication deficient, making them generally very safe. It has a broad host range, and critically for diseases affecting long-lived cells like liver hepatocytes, AAV vectors can efficiently transduce non-dividing cells. Importantly, AAV vectors typically elicit only a very low level of immunogenicity, particularly towards the vector capsid, the protein shell, allowing for persistent, stable, long-term gene expression. Furthermore, researchers have discovered and developed different AAV serotypes versions with different capsid proteins that exhibit distinct tissue tropisms. Serotypes like AAV5 and especially AAV8 show a very strong natural propensity to target the liver after intravenous injection, making them ideal for liver-directed therapies like Crigler-Najjar.
That sounds almost perfect. Are there any limitations with AAV, given that impressive list of advantages?
Like all vector systems, AAV does have some limitations. Its major constraint is a relatively small packaging capacity. Its genome is only about 4.7 kilobases long, which limits the size of the therapeutic gene cassette, the gene plus necessary regulatory elements that can be packaged inside. Luckily, the UGT1A1 gene fits. Also, similar to adenovirus, the AAV genome largely persists in the nucleus as an episome, an independent circle of DNA, rather than integrating into the host chromosomes. While this is generally considered a safety advantage as it minimizes the risk of insertional mutagenesis, it means that if the transduced liver cells divide, the episomal vector genome might be diluted out over time, potentially leading to a gradual loss of expression in a growing liver, though this is less of an issue in the stable adult liver.
So episomal persistence safe, but maybe not truly permanent if cells divide. What have the results shown for AAV in treating Crigler-Najjar syndrome in the gun rat models, despite that?
The therapeutic potential of various AAV serotypes carrying the UGT1A1 gene has been thoroughly tested in gun rats, often with very impressive outcomes. And the results reported have been truly significant, perhaps even exceeding the longevity seen with HD8 in some studies. For example, delivery via the portal vein, a major vessel entering the liver, of a specific AAV serotype vector, SAAV1 carrying the human UGT1A1 gene, was reported to achieve a stable, lifelong correction of the jaundice in gun rats, with about a 70% reduction in serum bilirubin levels maintained for the entire duration of the animals' lives. This demonstration of potentially lifelong efficacy after a single treatment makes AAV vectors an extremely exciting prospect for providing a durable, perhaps permanent cure for Crigler-Najjar syndrome, potentially freeing patients entirely from the burden of daily phototherapy or the need for liver transplantation.
What an incredible journey we've been on, really diving deep into the world of genomic medicine and the gene therapy program. We've navigated the sheer complexity of cancer, exploring these ingenious ways scientists are, re-engineering viruses, building hybrid RNA, DNA molecules, even shielding healthy cells from chemo's harsh effects. and then seeing how these groundbreaking approaches are being meticulously applied and refined for devastating genetic diseases like Crigler-Najjar syndrome. It's really inspiring.
It truly is. And it becomes clear, doesn't it, that gene therapy is aiming for something much deeper than just treating symptoms. It's fundamentally about understanding and attempting to correct the underlying errors in the very blueprints of life. Now, challenges certainly remain big challenges. Achieving consistently efficient and safe gene delivery in vivo, managing potential immune responses to the vectors or the therapeutic proteins, ensuring long-term durability. These are all areas of intense ongoing work, but the progress, especially over the last decade or two, has been undeniable, and the promise is just immense.
We really do seem to be standing on the cusp of a medical revolution, moving towards an era where personalized genomic interventions could genuinely transform healthcare as we know it, getting closer to not just managing but truly curing diseases at their genetic source.
So the big question remains, what does this all mean for us, for the future of health and medicine? As these gene therapy technologies continue to advance and potentially become more mainstream, how might they reshape our very understanding of health, disease, longevity, maybe even what it means to be human? And perhaps just as importantly, what are the ethical, societal, and economic conversations we need to be having now proactively as these powerful technologies become more widespread and accessible? There's a lot to consider beyond the science itself.
That's a perfect thought to leave our listeners with. Thank you so much for joining us on this deep dive. We hope this exploration into the intricate world of genomic medicine and gene therapy has given you a newfound appreciation for the power of science and the exciting, complex future of healing.